My name's Eleanor and I'm delighted to have the opportunity to tell you about our work on developing sonodynamic therapy as a treatment for pancreatic cancer. I'm presenting on behalf of a large team collaborating between the Universities of Ulster and the University of Oxford. And I also need to declare that John Callan, Tony McHale and myself are co-inventors on a related patent application and co-founders of a spin-out company. As you will probably be aware, pancreatic cancer has one of the worst prognoses of all cancers, and treatment outcomes have not improved for over 50 years. Due to its typically being diagnosed at a late stage, tumours tend to be large, poorly vascularized and hypoxic, and thus resistant to treatment. Patients are also typically unable to tolerate aggressive radio or chemotherapy. Sonodynamic therapy offers a means of significantly reducing systemic toxicity compared to conventional chemotherapy because the drugs are activated by ultrasound exposure, and so treatment can be localized to the tumour. One challenge, however, is the need for sufficient oxygen to be present for the drugs to work. To address this, we developed a microbubble drug conjugate, enabling oxygen to be co-delivered simultaneously with the drug. In our initial pilot study, we compared the effects of oxygen-loaded bubbles and sulfur hexafluoride-loaded bubbles in a mouse model of pancreatic cancer. We found that the oxygen bubbles substantially increased tumour response. But whilst these results were encouraging, there were many research questions that needed to be addressed. First of all, the mechanism underlying drug activation. The drugs used for sonodynamic therapy were developed originally for photodynamic therapy, and it was not known why ultrasound should have the same effect as light. Consequently, there was considerable skepticism as to the validity of sonodynamic therapy. For example, this was the Wikipedia entry. And luckily, thanks particularly to the fantastic efforts of Dr. Estelle Begin and Dr. Shamit Srivastava, we were able to at least partially unlock the mystery and demonstrate that the activation process was actually the same as for photodynamic therapy, but the light was generated by cavitation through sonoluminescence. And this is very exciting because it means that we can exploit all the benefits of photodynamic therapy, but at much greater tissue depths. Our second question was whether there was any advantage in combining sonodynamic therapy with low doses of conventional chemotherapy drugs. So we created a combination therapy bubble carrying both the sonodynamic therapy drug and gemcitabine, small molecule chemotherapy drug, and repeated the experiment. And we saw that there was indeed a statistically significant improvement in tumour response with the drug combination. Now, crucially, up to this point, all of the results I've shown used an intratumoral injection, which clearly isn't very translatable. So we had to make some modifications to improve the stability of the bubbles so that they could be given intravenously. Fortunately, we were able to do this and generate a similar therapeutic effect in the same tumour model, but now using intravenous administration. And very importantly, we saw no statistically significant difference in the weight of the mice between those that received the bubbles and the untreated controls, whereas those that received the equivalent of a standard gemcitabine treatment showed substantial weight loss. So we're now in the process of developing this approach for clinical translation. We have a protocol for manufacturing the bubbles to GMP standard and are raising funding for a first-in-human trial in patients with borderline resectable pancreatic tumours. Finally, I also wanted to share some of our recent data that we're very excited about. These were experiments in immunocompetent animals with 2 tumours and using microbubbles with just the sonodynamic therapy drug. Only 1 tumour was treated with ultrasound, but a response was seen in both animals that seems to indicate stimulation of an immune response. And when the experiment was repeated with an immunotherapy drug, there was an even larger effect. And just to note, all of these curves here are for the tumour that was not exposed to ultrasound. So to conclude, we believe that sonodynamic therapy is a very exciting approach for cancer treatment. We have shown that its efficacy can be increased both by the use of oxygen microbubbles as carriers and also by combination with low doses of gemcitabine. By modifying the microbubble formulation, we can enhance stability, and also, though I'm sorry I didn't have time to talk about it, facilitating targeting both through the use of antibodies and magnetic fields. And finally, we have preliminary evidence of immunostimulation that could potentially be useful for treating metastatic disease.